Good afternoon and welcome to the 18th Scientific Week of Faculdade de Medicina de Campus. Thank you for joining us.
Aí a gente está vendo aqui.
Ok. Oi. Por enquanto não estou não, tá você lá. Ó. Ah, sim. Good afternoon and welcome to the 18th Scientific Week of Faculdade de Medicina de Campus. Thank you for joining us live on Zoom for today's roundtable meeting, Pathogens Without Borders, a One Health Perspective on Fungi, Bacteria, and Protozoa. I'm Dr. Parisi, a former medical student of Faculdade de Medicina de Campus, a Brazilian board-certified plastic surgeon, medical doctor certificate in the US, finished a research trauma fellowship at the University of Miami and currently in training to obtain my U.S. medical license. I would like to sincerely thank you, the Faculdade de Medicina de Campus, Professor Alaide, and Ms. De Souza, our research coordinator, for the kind invitation to moderate this important roundtable. Today, we are joined by three outstanding speakers. Dr. Alves will be the first. He is a pharmacist with a master and doctorate degree in molecular and cell biology from the Oswaldo Cruz Foundation in Brazil, with part of his PhD conducted at the University of Liverpool. He completed a postdoc research fellowship at the University of California in Berkeley and served for 2 years as a visiting associate professor at the University of California, San Francisco School of Medicine. The second speaker is Dr. Belize, PhD in parasitology and ecology, currently a research associate at the Liverpool School of Tropical Medicine in UK. She has extensive research experience in Africa and the UK with a strong background in medical entomology and implementation research. The third one is Miss Humana, researcher at Universidade Estadual do Norte Fluminense with a strong focus in plant pathology and entomology, dedicated to advancing research and development in these fields. Each speaker will present for 20 minutes. Please hold your question until the end of presentations. We hope this roundtable meeting brings new ideas and share knowledge. Thank you again for being here. Dariel? Belize que está aí, né? Eu vou começar com ela.
Fala no microfone que ela ouve, gente.
É que eu não... Dr. Belize?
Professora,
Dr. Belize, could you please start your presentation? Can you hear us?
Give me a few minutes to share my screen.
Thank you, Dr. Belize, for your presence with us today.
Sorry, excuse me, I need to remove the subtitle. Give me a few minutes, please.
No problem. You have 20 minutes to present, Dr. Belize.
Yes. Okay.
Hey, Dr.
Let me
Belize,
show my face
welcome.
before starting.
Is everything okay now?
Good afternoon, everybody. I am Patricia Dumbe-Bellis. It is an honor to be amongst you, and I will try to share my expertise and what I have currently done and what I've done before in the past. And I would like to acknowledge the organizing committee to invite me and to be part of this panel. Thank you. So without further ado, we are going to start. Allow me to switch off my video before. Okay. Are you able to see? It's not okay.
I can see your slides.
Okay. I don't know how to remove this one. Okay. Sorry, I was not able to remove those subtitles. I hope that it will be okay. Okay, today my talk will be mainly focused on how to integrate the One Health concept for malaria elimination. And thank you, Patricia, for the small introduction. And you all know my background. I am mainly a medical entomologist, and I have been mainly work on vector-borne disease control since I am undergraduate. Then I moved at the Liverpool School of Tropical Medicine to conduct a project in order to control malaria vector in Ethiopia.
Impressive.
Yes, it was working. Okay. Malaria, or as a student, medical student, you all know malaria. Malaria, it is a parasitic disease, and actually across the world, it remains one of the most pressing public health challenge. And we, in low and middle income country, specifically in Africa, urban malaria is a growing threat. We experience in Africa, there's a lot of poor management of waste in many urban settings, an increase of informal settlement, poor drainage of water, as you can see in the different picture. Also, there are the presence of swampy area. All those factors lead to the proliferation of vectors, such as mosquitoes and flies and other vectors, which are responsible of vector-borne diseases like malaria, dengue, and other arbovirals. The main vector control of malaria relied on the use of long-lasting insecticide-treated net, indoor residual spraying. And unfortunately, those core tools are facing some challenges, namely the increasing insecticide resistance, the behavioral adaptation of vector, and also those environmental degradation due to household human construction in the city, which allow the proliferation of water body, increasing the density of mosquito among the communities. The One Health approach appears as a strategy to enhance malaria control and support long-term elimination efforts. And an holistic view of that approach in malaria control emphasizes the relation between human, animals, and environmental health in combating malaria. Also, there's a need of collaboration across different sectors because malaria it is a kind of dynamic disease. It affects the human, it's linked to the human, to the environment, to the vector, and all those factors need to be taken in account. That's why in the One Health approach, we need to increase the collaboration between the human health sector, the veterinary, the agriculture, social science, and also environment to get an effective malaria elimination strategies. Also, the benefits to implement these strategies to prevent all those vector-borne diseases rely on the setup of multiple determinants of health in a simultaneous way. Malaria, despite all those, malaria challenge in applying One Health to malaria relied in the coordination and resources allocation amongst those different sectors. Okay, if we want to have a look on malaria vector control from a One Health lens, we can think about targeting larva habitat, creating by agriculture practice, livestock watery point. Also, we can think about mapping the different breeding sites across the urban settings, set up genetically modified mosquito, and also integrated vector management. There are many different measures for the integrating vector management. It could be the combination of the core interventions such as mosquito net or indoor spraying with larva control, or the combination of those core tools with genetically modified mosquitoes or spray spraying, and there are other many control tools to manage the vectors. As I mentioned earlier, I have been mainly work in Africa and specifically in Cameroon and in Ethiopia. Across the low and middle income country, we have different kind of malaria vectors. In Cameroon and Ethiopia specifically, in Ethiopia, we have the invasive Anopheles stephensi. In Cameroon, the main malaria vector is Anopheles gambier, as you can see in the picture. And in the urban settings, we have different kind of breeding sites. It can be around the agricultural area in the lowland city of the city. It could be right after a flood or a huge rain, or it can be due to human construction. The landscape in Cameroon and Ethiopia are really different. Okay, in those studies, we try to control the larval stage of mosquito by using some product called larvicides as additional control tools to mosquito bed net. We use two products. In Cameroon, we use the Vectomas, a larvicide, a biolarvicide made up of Bacillus thuringiensis and Bacillus sphaericus. This larvicide was applied manually in natural breeding sites, as I presented before, the small spool and also those standing water bodies. So the application was conducted every 14 days. In Ethiopia, the landscape is different. It is a dry area, and people here used to store water in large containers. They call it beaker. And in that field trial, we apply another kind of larvicide called Sumilav 2MR. This Sumilav is made up of pyriproxyfen. It is an insect growth regulator. It was also applied manually, but at the difference to the Vectomas, this manual application was done once in those containers with a fortnightly monitoring. What we got from those trials, and as you can see in that picture, before the intervention at the baseline survey, the density of vector were high in the treated and the control area. After the implementation of the larvicides, you can see that the density reduced in red. It reduced in the treated area. And the result clearly show that the product has a huge impact in the reduction of Anopheles gambier density, the larva density, as well as the adult density. More information can be found in the paper sites below. In Ethiopia, this paper was just submitted. We find a 100% emergence inhibition rate in the treated containers, because these larvicides act like an inhibitor, growth inhibitor. And also, we record like the impact was really effective against the late stage of Anopheles stephensi. From those trials, we clearly see that the implication of community, it is a must. So that's why we recommend to empower community for effective malaria intervention, because it's the community who are the one who look after your product. They are the one who coordinate the implementation in the different breeding sites. So that's why there's a need to promote health initiatives that facilitate effective malaria intervention and mobilize community resources. Also involve residents, the local community in the decision making. We have to train them. We have to engage them more. They have to collaborate in order to lead the implementation of the different products on the ground. Also, there's a need to monitor and integrate the data, different data from the human cases, entomological surveillance, and also ecological comportment. Also, while mapping the breeding site in a city, it is a kind of risk prediction. If we proceed with a wide land, a wide mapping of breeding site, we are able to target the breeding site of mosquito, apply the product, involve the community. On that way, we are sure that the surveillance is really effective. And also, that One Health platform is really important to share the data between the different sectors. It could be the health sector, the environmental sector, agricultural sector.
Dr. Belize, you have 3 minutes remaining.
Yes, almost. Okay, the main challenge in that One Health implementation are mainly focused on the institutional silo because those sectors operate independently, and also there is no fund allocated to combine the research together in the different sectors. To move forward, we are planning to integrate One Health into national malaria. We recommend to integrate One Health into national malaria strategies in the different endemic countries. We also recommend for those governments to invest in intersectoral collaboration. There is also a recommendation based on innovation, put in place some new tools in order to control the disease. To conclude, malaria elimination is possible, but requires system thinking. One Health bridge the gap between biology, environment, and community. Sustainable success depends on cooperation and integration. With that, I would like to thank you for your kind attention. We can hear you. Patricia, you are mute.
Sorry, my bad. Thank you, Dr. Release. Your presentation was very insightful. It was a pleasure to learn with you. I'd like to ask you a question.
Yes, go ahead.
Okay. So, how has your experience in Africa and the UK shaped your view on global disease control?
Okay, thank you for your question. I think it is, I realized that while working in the two environments, I realized that it's two settings completely different.
Mm-hmm.
Yes, talking about the presence of a system, parasite vector system. The disease we found in Africa country are not the same in European country, in UK specifically, and also even the management of those diseases, how the government set up teams to control those diseases. I mean, in Africa, it's not yet developed. The implication of the community, the government, the stakeholder is not really high. They need more support in order to set things well, coordinate, and get successful result to control those diseases. It's not only malaria, it's vector-borne disease in general. So, I mean, this is my first insight to your question.
Thank you very much. Wonderful experience. So I'd like to open the question to the audience. Any question from the audience for Dr. Belize?
Okay.
I cannot hear.
Can you hear me?
Yes.
Okay, thank you. The name is Ifeoluwa, doctoral student from UNFI. Nice presentation from Dr. Belize, Belize. I want to just ask, I want to know if, prior to the research, there was screening on the index for mosquito in Cameroon. Is it only Anopheles gambiensis that was identified? Possibly that is responsible for malaria in Cameroon. So I want to have an idea of if that was the only parasite that was observed to cause malaria in Cameroon.
Okay, thank you for your question. I mean, I just mentioned, to go straight, Anopheles gambier is not the only malaria vector, but it is the main species involved in the transmission. We have different complex of vector, and we have a lot of publication. I just present, I put Gambier forward because it was my vector of interest. We have Gambier, we have, within the complex Gambier, we have Gambier, Sensus lactus, we have Arabiensis, we have the complex, the group Nestus, and all those vector breed is like they spread across the country depending on the geographical location. Because if you move in the North Cameroon, for instance, you will see that the main vector is Arabiensis. In the central region of Cameroon, the main vector is Gambier. In the coastal region, we have Merus, Meras. So there's a large panoply of vector in Cameroon.
Okay, thank you very much. Well, finally, the reason why I asked is there was a particular research which I was part of that was conducted in Nigeria. And we discovered that for majority of, we were trying to evaluate, and we discovered it was not just Gabiensis that was present in that location. We even just used a location. And we discovered there was Arabiensis and other vectors for mosquitoes. So in order to carry out a One Health strategy or approach, it's just a recommendation for the team. I understand you talked about limitation in terms of finance or capital intensive. I would want to recommend so that it will be good that as we make these approaches, we screen through the whole vectors that are responsible for malaria. And if resources are not readily available, you could do a retrospective study, probably get resources from the epidemiology unit of the university, of any university that has done researches on mosquito or malaria parasite before now, and you'll be able to screen to be able to see if this One Health approach could be utilized effectively, than focusing mostly on maybe just a vector for the experiment. Thank you.
Yes, thank you. This is a good and great recommendation. Thank you.
Any other question from the audience? Thank you very much, Dr. Release. Your
You're welcome.
experience is impressive.
Thank you too.
Patricia, there isn't any question from the audience here.
Okay.
Is there any question, guys? No? No, there isn't. We can move on.
Okay. So let's go move on to another speaker, Miss Anwana. Miss Anwana, could you please start your presentation?
Hi, good afternoon, everyone. I'm Diana Wana Afia, a Nigerian here in Brazil to do my doctoral study in using endophytic fungus to control some pests of agricultural plants, and not only the pests, also pathogen. I am pleased and very opportune to be here to present on this wonderful topic titled Harnessing Endophytic Fungi, Bridging Pest Control and One Health in Perspective. Okay. Before I start, I just want to tell you that this wonderful fungi have been growing with this plant, and they have not caused disease in the plant. Basically, they grow as a mutualistic kind of relationship with the plant, thereby protecting the plant, and not only protecting the plant, they have also revolutionized the pest control strategy in controlling pests that comes to this plant. So I am very happy to be presenting on this topic. The next slide. Where?
Yes.
See. As we face challenges of sustainable agriculture, conserving our environment, and maintaining public health, we see that these issues are deeply interconnected with each other, and they reflect a complex web of interdependence. Now, in recent decades, chemical pesticides have been used to control pests and pathogens. And because of that, the overuse of this pesticide has caused numerous environmental concerns, not only to the environment, but also to the health of the consumers. Now, its indiscriminate application has also led to contamination of our soil, our water, our air, contributing to long-term environmental degradation. Okay. Now, this chemical has disrupted different ecological balances. If you look at what this picture is saying, the pest is asking the farmer that, now you are using the pesticide on me. Are you also going to use the face masks while consuming your vegetable? That is a very vital question. But we just consume our plant. We just consume our plant not knowing the kind of chemicals that have been used on this plant. Now, this cumulative effect of these practices have underscored the urgent need for a more sustainable and ecologically sound pest management strategies. That's why we're making use of our endophytic fungus, trying to harness them to control pests and diseases on the farm. Why? Because these endophytic fungus are very environmentally friendly. They are no risk to the environment or to the health of an individual. Now, these fungi, as I said earlier, they live harmlessly within plants, play vital roles such as protecting the plants and also contributing to the well-being of humans. Now, what are these endophytic fungus? They are fungus that colonize plant tissue internally from the root to the stem to the leaves, living inside the plant, protecting the plant while also benefiting from the plant. The plant provides shelter for them, and they too help the plant by preventing any attack of incoming pathogen or pest. And they can be transmitted from one plant to the other, or from one plant to the seed. And when we talk of one plant to the seed, that is vertical transmission, but from a plant to another plant via spores, where the spores from one plant out from the endophytic fungus goes to another plant. We call that horizontal transmission. Now, these endophytic fungus are very diverse. They are widespread. They can be found in the Ischomycota, the Basidiomycota, and the Micromycota. And they have been isolated from different plant species via different methods have been employed to get these endophytic fungus from this plant to study them further. And also, this endophytic fungus deploys several mechanisms in protecting the plant from an incoming pest or a pathogen. And this mechanism can either be direct mechanism or an indirect mechanism. Now, for the control of pests, using how this endophytic fungal works is that for the direct mechanism of control, it releases metabolites in the plant. The endophytic fungus releases metabolite which can either affect the insect, or it can deter the insect away from the plant, or it can induce the plant itself to produce volatile organic compounds. These organic compounds attract other insect parasite, prey, to come and feed on the incoming insect. It's just like I'm inviting my enemy to come and attack the person coming to my house uninvited. And while the indirect mechanism involve the induction of the plant system to produce this volatile emission, thereby protecting the plant further. And for mechanism for the pathogen control, the direct mechanism is the mycoparasitism. When we say mycoparasitism, it involves when this endophytic fungus inside the plant attacks the incoming pathogen, feeds on this incoming pathogen, or via competition, whereby it limits this pathogen from accessing food, from accessing water, thereby starving the incoming pathogen, which eventually dies off, or via antibiosis. Here, the endophytic fungi releases some metabolite that in turn affects the incoming pathogen and kills it. While the indirect mechanism of control, it can induce two plant defense mechanisms, either the systemic acquired defense or the induced systemic resistance in the plant, thereby preventing the proliferation of this pathogen within the plant tissue. Okay, these are an example of an endophytic fungus in action. Beauveria bassania was incorporated. This is showing a tomato plant without an endophytic fungus. If you look at the leaf, you can see that the plant was visited by the leaf miner, Tuta absoluta. You can see mines on the leaves. The fruit is looking terrible because the presence of this fungi was not there. But the next slide is showing us a plant colonized by endophytic bacteria, Beauveria bassania. Now you can see how the plant is looking very healthy. Why? Because this fungi is inside the plant, protecting the plant from any pest or pathogen. Okay. Now, the One Health Frame Network emphasizes the interconnectedness of human, animal, plant, and environmental health. How do these endophytic fungi fit into this One Frame Network? By reducing the use of chemical pesticide. Making use of this endophytic fungus reduces the residue of chemical pesticide on our food, thereby making our food safe for consumption, minimizing environmental pollution, and decrease harm to the beneficial insects such as the bees, the butterfly that pollinates our plants. They enhance food, thereby lowering incidence of foodborne illnesses and reduce risks associated with pesticide exposure. Also, reducing chemical residues in food enhances human health and help mitigate microbial resistance. And according to a Canadian pesticide control, they said some of these pesticides used on our agricultural plants have resulted to various either the short-term health issues or the long-term issues such as dizziness, skin irritation due to exposure of this pesticide. Then for the long-term issue, they mentioned over-accumulation of these chemical residues in our body had led to some cancer cases, birth defects, and so on. And environmental benefit of this endophytic fungi includes improved soil health, preserve our biodiversity, mitigate pests and resistant development from the use of chemicals to control these pests, which are very crucial for agricultural systems. Now, owing to the numerous benefits of this fungi, endophytic fungi, it has been developed into an entomopathogenic fungi to control pests in a conventional method instead of making use of your pesticide chemicals. Now, as EPF, entomopathogenic fungi, they are a group of naturally occurring fungi that kill insects on contact. Unlike chemical pesticides, they offer a biological means of pest control, making them part of an integrated pest management program. And some common examples that have been made use of so far is the Beauveria, the Metahaesium, the Licancilium, and the Ischia. Even though Trichoderma is one of the endophytic fungi that has been explored for its entomopathogenic properties. Now, the mode of action of this EPF, how does it work? First, the spore of this fungi just needs to come in contact with the cuticle of the insect. When it comes in contact with the cuticle, it adheres to the cuticle of the insect. From there, germination takes place, and from germination, it penetrates the insect, occupies the internal part of the insect, leading to the death of the insect before the mycelium grows out of the insect, covers the whole insect, then the spore is being released externally. Now, examples of EPF that we've been making use of in the lab at WEMFI, we have Beauveria bassania and Metahesium anisophil. They are the two most used fungi endophyte right now. Now, this is their mode of action. The first is the action of the Beauveria bassania, while the next is the action of Metahesium on this insect. Now, they produce insecticidal toxins that kill insect pests following the infection. Now, this is the mode of action, the life cycle of what happens when these entomopathogenic fungi come in contact with an insect. Now, they are widely used in integrated pest management. Now, how are EPF being used as endophyte? How do we incorporate them as endophyte into plants for protection? Now, it involves inoculation techniques that is designed to facilitate fungal entry, its establishment within the plant, and systemic spread within the plant to proffer protection for this plant. Now, the common inoculation method of EPF in plants include seed coating. Here, the seeds are being coated with spores of this entomopathogenic fungi left for a time before planting, and this grows with the plant, or via root dipping. In this process, the plant is grown. After a few weeks, it's been uprooted and placed in a suspension of spore suspension of this entomopathogenic fungi for some time for the root to absorb this endophytic fungus before replanting it, and then watch out for its colonization within the plant, or via soil drenching. Here, the suspension is used to irrigate the soil, thereby giving the root access to this entomopathogenic fungi to be absorbed and colonize the plant, or via foliar spray, where this entomopathogenic fungi is turned into suspension and used as spray. And this, when they are being sprayed on this plant, the plant absorbs this entomopathogenic fungi into it, and then the colonization of this fungi takes place in protecting the plant further. Sorry. Practical applications we've had.
Excuse me, sorry. We have 4 minutes remaining.
Okay. We have current success on the use of this entomopathogenic fungi to applying it in the plant. Here, from this, you can see colonized with Beauveria bassania. You can see how the tomato plant is growing very well, while the control is looking like a plant starved from malnutrition stuff, if you can say that, and the fruits are looking great. The method for colonization of this was via the seed. The tomato seed was colonized using Beauveria bassania. Now, the application of this fungi is a little bit limited due to some factors such as temperature, humidity, light, ultraviolet radiation affects the how do I say this in English? It affects how the duration or its effect in application. this limits its effect on the plant. If this temperature, humidity, light, and UV radiation is increased, it limits how this EPF plays its roles in protecting this plant. And also challenges of the application of this EPF lies in ensuring consistent efficacy under diverse field conditions and overcoming regulatory hurdles for its use as biocontrol, and developing formulations that are easy to apply and affordable. Potential risk and consideration. While this endophytic fungi is very promising, there are some required understandings of its ecological impact, its potential for non-target effects, and regulatory hurdles. For example, the safety of fungal metabolite must be carefully evaluated because some of these metabolites might be harmful to the environment. And future research frontiers, genetic engineering to enhance fungal virulence and its specificity, microbiome engineering, that means combining multiple endophytes for synergistic effect in integrated pest management, then system biology approach in understanding the plant, the endophyte, the insect interaction. In conclusion, endophytic fungi are a treasure trove for natural pest control agents, and they offer a very wide, sustainable, eco-friendly, and alternatives to chemical pesticide. Their use aligns with the One Health paradigm, thereby promoting environmental integrity, protecting our food, and also human health. The path forward involves target research, fostering multidisciplinary collaboration, and informed policy making. Thank you.
Thank you very much, Mrs. Awana. Your presentation was very impressive, very instructive. Can I ask you a question?
Sure, yeah. Yes.
Okay. About your research in the community, is your experience, how can agricultural or entomological research improve public health outcomes in rural or underserved communities?
Okay, thank you very much for the question. Basically, we're trying to talk to the farmers to tell them that these fungi are very safe for the environment, even though these farmers are already used to chemical pesticide because that has been their first approach to controlling this pest. But right now, the endophytic fungi is very safe. It's environmentally friendly. It doesn't pose health to animals, plants, or humans. It's safe and it can be used to control pests. So it's a collaborative kind of training to make them get used to the idea that these fungi can be used on farm to protect the plants against pests.
Mm-hmm. Sounds amazing. Thank you very much.
Thank you.
I'd like to open question to the audience. Any question from the audience?
I do. I have a question.
Okay.
Ms. Uwana told us about the pathogenic fungus for the insects. And, well, my biggest worry would be with that, that the fungus would also attack the predators of the pests, like aphids and ladybugs. And you mentioned also the possibility of genetic engineering for them to be more specific. WENF has a somewhat big bioengineering department. Are they also working in developing more specific strains of the fungus?
Right now, I don't know, but We have not had that for now, but we're working towards that.
Okay, thank you.
Any other question?
Pessoal, se quiser, pode perguntar em português também, tá? Alguma pergunta? Pode ser em português, pode ser em inglês. No. Everybody's shy here today. There is a question, a comment.
Maybe Lucas will help me translate in Portuguese. A follow-up to... Okay. So a follow-up to the question that was asked by Dr. Patricia. I for a person, wants to believe that what One Health wants to achieve is a multidisciplinary approach to addressing global issues. And if we must be able to carry out these global challenges in a manner that we find lasting solutions, then every discipline that is involved has to participate even more. Take, for example, as medical doctors, when you have patients that comes to you and you want to review the patient, taking their prognosis, and you discover that the person most likely is a farmer, or the person is most likely staying in a settlement, that there are so much farming activity, it is important for us to make this awareness about how that they get to use a lot of chemical pesticide, and that the residues that they get to have affect them in a way, and it's telling on their health. And so most of the times, it is, if we must be able to allow One Health thrive, because as it stands, it's still a growing discourse. It's still a discourse that is not yet pronounced. It's still very, very young. It will be important that medical doctors, pharmacists, nurses would also try to, aside from administering medical counsel, should also try to counsel as many persons that probably has their profession as farmers, that, oh, have you heard of this very particular control mechanism, biological control mechanism you can use to control the pests in your field instead of using chemicals that could be dangerous to your health. So it is very important that we take this. That is why a conference like this is put together, so that we can be informed. Lastly, take for example in Nigeria, there was a time that this variety of sweet potato, what we call batatas, here in Brazil, was developed, orange flesh sweet potato. At some point, it was recommended to farmers and even people who comes to the hospital because of the glycemic level, the glycemic index compared to the conventional sweet potato. That, oh, if you don't want to use azúcar, that is sugar, you can basically use your orange flesh sweet potato, which is quite healthy for you. But we discover that most times recommendations are usually on drugs. Meanwhile, there are a lot of fruits that can be administered by doctors, recommended by doctors, nurses, pharmacists, to allow people to take just to improve their health. So if we must make One Health thrive, we should be able to participate as a discipline. Thank you.
Thank you for the question. Any other question from the audience?
No questions, Patricia, here.
Okay. Thank you to all our speakers. We appreciate your participation. Your presentation were amazing, fantastic, very, to me, it was like an eye-opening. So So, thank you, Faculdade de Medicina de Campos, for the opportunity to be a moderator during this meeting. Thank you, Dr. and Professor Alaide and Ms. De Souza. Thank you, the audience, and I would like to invite you to see you in the future sections during the 18th Scientific Week of Faculdade de Medicina de Campos. Thank you very much.
Patrícia, it was an honor to be here with you. Thank you for coming, for sharing, and thank you, Diana, and the all people here. Gostaria de aproveitar, comentar com vocês. Patrícia é egressa da Faculdade de Medicina. Você chegou a falar sobre você, Patrícia?
Cheguei.
Conversou com eles, né? Muito obrigada pela parceria, obrigada a todos os alunos. Gostaria de reforçar, Dr. Fábio teve um imprevisto nos Estados Unidos, ficou muito chateado, não pôde estar conosco hoje. Não faltará oportunidade. Muito obrigada aos alunos, a todos, Patrícia, everybody, thank you.
Obrigada, professora. Bom, eu gostaria, em português, só de fazer um convite para os alunos da Faculdade de Medicina de Campos, porque, assim, para mim, essa faculdade sempre abriu nossos olhos para voar para o mundo. Nós fomos ensinados, desde o primeiro ano, a ser cidadãos do mundo e não de Campos, e nem do Rio de Janeiro, e nem do Brasil. Então, qualquer pessoa que tenha interesse em fazer residência fora, tem interesse em fazer um observership, um externship, um clerkship, e tem alguma pergunta, eu estou aberta para ajudar, para tirar as dúvidas. É só me seguir no Instagram e me perguntar, e eu estou lá para ajudar, porque para mim está sendo uma experiência muito legal. É um passo grande, são muitos obstáculos, muitos steps, mas está valendo a pena. Hoje eu sou um médico certificado aqui nos Estados Unidos e estou em training, mas estou caminhando aí para ter minha licença, e eu estou aberta para ajudar. Qualquer dúvida é só me seguir no Instagram. Tudo bem? Muito obrigada pela oportunidade.
Thank you, Patrícia.